We are going to go through the WAKISHA joint mock papers of 2026 A-Level Biology. A bean seed multiplication program operating in the highlands of Kabale district expanded production to lower altitude areas in an attempt to increase seed availability. After several planting seasons, farmers observed reduced crop growth and poor seedling establishment. Investigations revealed that the lower altitude sites experienced high daytime temperatures and intense sunlight. Soil assessments also showed declining mineral availability due to continuous cultivation without adequate nutrient replacement. In addition, many harvested seeds failed to germinate even under favorable planting conditions. Researchers observed several plant responses. Seedlings growing under shaded conditions elongated their stems rapidly and produced broader leaves. Some mature plants shed older leaves during prolonged stress, while seeds remained dormant until environmental conditions improved. This means we are going to analyze and evaluate. Let us analyze the table of the findings: increased photorespiration, reduced chlorophyll, and ATP production, and seed germination. Task Part A. Analyze the observed changes in the table lead to reduced crop growth and poor seedling establishment. Part B. Evaluate the effectiveness of the plant responses in maintaining survival under the prevailing environmental conditions. We are going to analyze and evaluate. Before, in all bookshops in Uganda, get yourself A-Level Self-Study Biology textbook now. Analysis. What happened, why it happened, and how it happened. Increased photorespiration rate. Photorespiration increased from 18 at the highland site to 67% at the low altitude site. This occurred because high temperatures increased the oxygenase activity of rubisco, causing RUBP to combine with oxygen instead of carbon dioxide. Consequently, previously fixed carbon was released as carbon dioxide without producing sugars. This reduced the amount of carbohydrates available for growth and development, leading to reduced crop growth, reduced chlorophyll concentration. Chlorophyll concentration decreased from 92 to 48%. This occurred because continuous cultivation depleted essential mineral elements such as magnesium and nitrogen required for chlorophyll synthesis. Reduced chlorophyll lowered light absorption by chloroplasts, decreasing the amount of light energy captured for photosynthesis. Consequently, less glucose was produced for growth, resulting in stunted plants. Reduced ATP production rate. ATP production decreased from 95 to 44%, reduced chlorophyll concentration, lowered excitation of electrons, and decreased electron transport along the electron transport chain. Consequently, less photophosphorylation occurred, reducing ATP synthesis. ATP is required for carbon fixation, active transport, protein synthesis, and cell division. Therefore, reduced ATP production limited growth processes, leading to poor crop growth. Reduced seed germination. Seed germination decreased from 90 to 36%. This occurred because increased ABA levels maintained seed dormancy. ABA inhibited the synthesis and activity of hydrolytic enzymes required to mobilize stored food reserves and suppressed embryo growth. Consequently, many seeds failed to germinate even under favorable conditions, resulting in poor seedling establishment. Rapid stem elongation and production of broader leaves. Rapid stem elongation enables seedlings to grow above neighboring plants and reach areas with higher light intensity. Broader leaves increase the surface area available for light interception, enhancing photosynthesis under shaded conditions. Therefore, this response is effective in improving light capture and increasing the chances of survival. However, excessive stem elongation produces long stems with poorly developed supporting tissues. Consequently, plants become weak and are easily bent or broken by wind and rain. In addition, more assimilates are used for stem elongation instead of strengthening tissues and producing reproductive structures. Recommendation: proper plant spacing. Proper spacing reduces competition for light among plants, decreasing the need for excessive stem elongation. Plants receive adequate light while developing stronger stems and supporting tissues, thereby improving growth and reducing stem breakage. Shedding of older leaves. Shedding older leaves reduces the total leaf surface area exposed to transpiration. This lowers water loss and reduces the demand for mineral nutrients during periods of environmental stress. Consequently, the plant conserves available resources and survives adverse conditions for longer periods. However, leaf shedding reduces the photosynthetic surface area available for light absorption and carbon fixation. Consequently, less glucose is produced for growth, reproduction, and storage, resulting in reduced productivity and slower recovery after stress. Recommendation: application of organic manure and balanced fertilizers. Organic manure and balanced fertilizers improve soil nutrient availability and water holding capacity. Adequate nutrients support the maintenance of healthy leaves and reduce stress, allowing plants to retain a larger photosynthetic surface area and maintain higher productivity. Seed dormancy maintained by ABA. Seed dormancy prevents germination during unfavorable environmental conditions. By inhibiting embryo growth and delaying germination, ABA protects seeds from exposure to conditions that could lead to seedling death. Therefore, dormancy is effective in ensuring long-term survival of the species. However, prolonged dormancy delays germination and reduces the number of seedlings established during the planting season. Consequently, crop stands become poor and overall productivity declines. Recommendation: seed priming. Pre-soaking seeds before planting. Seed priming hydrates seeds and initiates metabolic processes required for germination without allowing full emergence of the radicle. This reduces the inhibitory effect of ABA, promotes enzyme activation, and enhances uniform germination, thereby improving seedling establishment. Thank you for watching.